I'm Hugh Mason, Associate Professor in School of Life Sciences and a member of the Biodesign CIVV. It's my pleasure today to introduce Mary Pardhe, who's a Ph.D. candidate in the MCB program. She joined my lab in the spring of 2017 and very quickly demonstrated her effectiveness as a researcher. In spring of 2018, she graduated after doing a very nice honors project on the production of a chimeric antibody targeting flavivirus envelope proteins. So she graduated with a BS in the MBB program and a minor in business, as well as a certificate in applied business data analytics. So she obviously has some interest in business. She started in my lab at the fall of 2018 as a 4+1 graduate student, but then the following year switched directly to the PhD program, which she's been working on now for the last couple of years. So she's finishing pretty early out. Well, I guess 3 years after the her BS. She also had finished a computational life sciences certificate program. And beyond that, she and another graduate student in my lab, Joe Hunter, have started a new company called Vaccina LLC, which will be using some of the technology that we've developed for recombinant immune complex. And that's going to be the main topic of her dissertation presentation today, entitled Plant-Expressed Vaccines, Enhancing the Recombinant Immune Complex Platform. So thank you, Mary, and take it away.
Thank you, Dr. Mason. Let me just close that. Okay, so can everyone hear me through the microphone? And is everyone able to hear me online?
Yes, I can hear you.
Cool. Thank you all for coming to my doctoral dissertation titled Plant-Expressed Vaccines, Enhancing the Recombinant Immune Complex Platform. This presentation will have 4 parts. First, I will present some background terminology and an introduction to immune complexes. Next, I will talk about how we can modify the recombinant immune complex to increase its versatility as a vaccine platform. Then I will talk about how we are studying the co-delivery of RICs with virus-like particles in order to gain a synergistic enhancement in immunogenicity. And finally, I will wrap up with a discussion of how we can use immunoinformatics in order to design better vaccines. The presentation will be about 30 to 35 minutes long, and I'd appreciate if you could save questions till the end, and then I'll answer all of them at once. So let's start with an introduction to recombinant to immune complexes. The first term that I will be presenting is antibody. Antibodies are specialized proteins that are generated by the immune system in response to a pathogen. Each antibody recognizes with high specificity a certain target. This target can either be a 3D conformational protein, or it is a linear string of amino acids, which are the building blocks of proteins. These epitopes are found on antigens. In general, an antigen is anything that produces an immune response, but in the context of vaccine development, an antigen is often found on the pathogen that we are trying to create a vaccine against, such as a virus. The goal of vaccination then is to present antigens to the immune system so that the immune system can mount a strong protective response. This response should be strong enough so that in case the host encounters a real pathogen, it is able to control the infection. Immune complexes are created when an antibody recognizes its cognate antigen. This finding, when it naturally occurs, is highly immunogenic since it stimulates many cellular pathways that leads to the generation of antibodies against that particular antigen. However, monomeric immune complexes, which are formed by an antibody binding to an antigen via its 2 binding sites, one on each arm, has been shown to provide an immune response against the antigen. However, there are some manufacturing challenges if you were to try to create a vaccine based on this immune complex. Some of these challenges include the necessity of first finding an antibody and a cognate antigen. This can be difficult to do, especially if you're trying to develop a vaccine against a novel pathogen. In addition, if you were to manufacture such a vaccine, you may have to produce antibody and antigen in different expression systems, which can first increase the cost, and any small changes to either the antibody or the antigen based on the expression can inhibit its ability to properly form complexes. Our goal then was to find a way that we could take all the benefits of immune complex formation without having to run into all these challenges. And this led to the creation of the recombinant immune complex platform. This platform has an antibody that is linked on its C-terminus to an antigen followed by an epitope tag. Earlier I talked about how every antibody binds to a certain epitope with high specificity. And by creating this recombinant immune complex, we were able to create these complexes that form when the RIC antibody binds to the epitope tags on surrounding RIC molecules. This forms these large clusters that have been shown to be highly immunogenic and able to stimulate all the pathways that we assume that normal immune complexes stimulate. So we decided to pursue this RIC construct further and study its ability to be used as a vaccine platform. So our goal with this project was to create an ideal vaccine platform that can be used for the efficient creation of vaccines against both existing pathogens and newly emerging pathogens. So what would be the characteristics of such an ideal universal vaccine platform? Well, first of all, it must be easily modifiable. In case a new pathogen arises or we need to create a vaccine against an existing pathogen, we need to be able to modify the platform conveniently and swiftly. Next, it should be efficacious, which means that it needs to be able to produce a strong immune response. The goal of vaccination is to produce an immune response, and ideally, we'd be able to produce this response in as few doses as possible so that we are able to efficiently get it to the people who need it. Next, it should have low production costs. If you were trying to create a vaccine against a novel pathogen, you want to make sure that it is cheap enough so that when we finish testing it, we're able to quickly scale up manufacturing costs and be able to distribute it to patients. And low production costs will also help us to get more equitable vaccine distribution. And as I mentioned before, we need to have quick scale-up potential. What we found is that plant-expressed recombinant immune complexes meet all these characteristics for an ideal vaccine platform. And therefore, we decided to study it further to figure out what are ways that we can do to enhance its capabilities as an ideal vaccine platform. Our first question that we asked is whether or not a recombinant immune complex is able to generate the same immune responses, similar or greater immune responses to naturally occurring immune complexes. And to do that, we designed the following experiment. As shown in part A, we first created an antibody called HSP8 that binds with high specificity to an antigen, GD6H. When we produce this antibody and antigen and mix them in solution, we create these naturally forming immune complexes. Each antibody will bind to 2 molecules of antigen, one on each binding arm. As a comparison, we also created this recombinant immune complex that has the exact same antigen, only displayed in our RIC platform. Once again, the RIC antibody binds to the epitope tag on surrounding RIC molecules and creates an immune complex. We then took these 2 constructs and tested them in a mouse immunization trial. And here are the results from that trial. The immunized mice with the exact same amount of antigen delivered either as an immune complex or recombinant immune complex collected blood samples after each dose and then measured the blood sample to test the level of antibodies that were produced against the antigen of interest, which in our case is GD, a small piece of the herpes simplex virus. In all 3 doses, we found that the RIC marked in green produced statistically significantly higher antibody titers when compared with the immune complex marked in red. Both of them were far above the control, which was a non-reactive phosphate buffered saline solution that was given to the mice to ensure that the results that we were seeing were due to our immunization and not due to infection of the mice themselves. These results showed that immunizing with recombinant immune complexes produces higher antibody titers than immunizing with immune complexes. This also showed us that our recombinant immune complexes were performing as we had expected, and then we decided to focus more on optimizing the platform. And that takes us to the second part of the presentation, which are RIC modifications and how they can increase its versatility as a vaccine platform. Once again, here is a schematic of the original RIC platform. We have an antibody that is fused on the C-terminus, which is the bottom part of the antibody, to the N-terminus, the top part of an antigen, followed by epitope tag marked in purple. Now, as I mentioned before, proteins are nothing more than three-dimensional structures that are formed by a linear string of amino acids that are then folded into a three-dimensional shape. When we notate these string of amino acids, we notate them in the orientation of protein synthesis, which means that they have an N on the start and a C at the end. However, there are some instances where we might not want to fuse an antigen in this particular orientation at the C-terminus of the antibody. These can include cases where the antigen that we are trying to mount on the platform has an inaccessible N-terminus. As shown in this picture over here, the N-terminus of this protein is marked in red. And in this protein, the Zika virus envelope, the N-terminus is actually buried inside the protein. So if we were to pull out the N-terminus in order to link it to the platform, as shown over here, we would be changing the overall structure of the protein. And that can be extremely detrimental to a vaccine because our goal is to present the antigenic epitopes to the host in as original a way as possible, because any modifications to the overall structure of the protein can lead to a change in the epitopes, especially the conformational epitopes, and that could skew the responses to a point where if the host ever encountered a real pathogen, they would be unable to properly react to it. Another case where you might not want to have a fusion in this particular instance is if we have a bunch of neutralizing epitopes on the N-terminus of the protein, and we want to make sure that it's more accessible to the immune system. So in order to resolve these instances, we decided to create a variant of the RIC platform. This is the original RIC platform once again, and this is our variant. In this variant, we changed the antigenic location to the top of the antibody, the N-terminus, instead of the C-terminus. For this experiment, we used the model antigen of the Zika virus domain 3, which is a small piece of the Zika virus. And we created two constructs. One had the original antigenic fusion on the bottom of the antibody, and the other was our modified RIC. Our question then was, first, do both of these constructs produce the same immune response in a mouse immunization study? One of our concerns was that the N-terminal fusion might inhibit the binding of the antibody to the epitopes on the surrounding RIC molecules. After all, the recognition site of an antibody are located on the arms, and if you were to fuse an antigen right there next to the binding site, it's possible that this could inhibit binding. So we created these two constructs and tested them in a mouse study. What we found after the terminal bleed is that both the antibody titers for the C-RIC and our modified N-RIC were the same, were very similar without any statistical difference between the two. We then followed up our study with a plaque reduction neutralization test, which takes some of the sera from the mice and mixes it with live Zika virus to test whether or not the antibodies produced by the mice in response to our vaccination are able to control the Zika infection. And what we found is that both the C-RIC and the N-RIC had similar results in the plaque reduction neutralization test. So these results showed that we were able to increase the platform's versatility to work with antigens that either need a C-terminus fusion or an N-terminal fusion. However, it's important to note that these results will be antigen specific, since we have found that fusing larger antigens to the N-terminus antibody does start inhibiting the binding and therefore causes smaller complexes. So these results have to be tested on a case-by-case basis. However, we have found that this is another variant that we can use in order to increase the versatility of recombinant immune complex. The next issue that we tackled is the inherent insolubility of the RIC platform. So in order to understand why insolubility is a problem, we first need to understand what solubility is and do a brief overview of how we produce these proteins in the first place. Solubility refers to a protein's ability to stay in an aqueous or water-based solution. And the way we produce these proteins is as follows. First, we start in the lab by creating a plasmid that contains the genetic blueprint in order to create our recombinant immune complex. We then take these genetic elements and put them inside a bacterial transfer vector that does the job of shuttling our bacteria from the lab to the plant cells. In this case, we use plants as a little factory in order to take the blueprints, create the protein, and then we purify the protein from the plant. Once we have our little bacterial vector that contains, or a bacterial system that contains our plasmid of interest, we infiltrate it inside plants, wait 4 to 5 days, and then harvest all the plant leaves and put them in a blender with a buffer. Then we grind up the leaves to produce a plant extract, and then we spin the extract at a high speed, about 15,000 times the force of gravity, in order to separate the extract into the soluble portion, which is this clear green liquid on top, and the insoluble portion, which forms a pellet at the bottom of the tube. This insoluble pellet contains molecules that are hydrophobic, that are not soluble, but it also contains large aggregates. And remember, the whole goal of a recombinant immune complex is to form these immunogenic clusters. The problem is, though, if they form two large clusters, they can all go to the insoluble pellet, leaving very little protein in the soluble portion. This is important because only the soluble portion is used for our purification strategy. We take the soluble portion of our plant extract and pour it down a specialized column that has these beads at the bottom. The beads trap all the RIC molecules from our plant extract, and anything that cannot bind to the beads flows straight through the column. Once everything is bound to the column, we add an acidic buffer which releases the binding between the RIC and the resin, and we're able to collect our purified protein. To understand whether or not our recombinant immune complexes were insoluble and how that was affecting our total protein yield, we did the following experiment. We created two constructs. The first is HL. HL is just a standard antibody. It does not have any antigenic fusions or epitope tags. We also created HLE. HLE does have an epitope that is specific to the antibody. So HLE is able to form immune complexes. So once again, the only main difference that we anticipate between these two constructs is the ability to form complexes. We then did a Western blot, which is an assay that qualitatively checks how much of a sample is in any given fraction. And as marked in red, if you were to compare HL and HLE, the addition of the epitope tag, which gives the construct the ability to form immune complexes, makes the entire protein, or most of the protein, go into the insoluble fraction. So once again, if you compare HL and HLE, you can see that HLE has a greatly reduced band. We then further studied these two constructs by doing the Western shown on the right over here. And this Western, we compared the soluble versus the insoluble fraction of HLE, the construct that can form immune complexes. And what we found is the addition of the epitope tag made a majority of the protein go to the insoluble fraction with only a little bit left in the soluble portion. Since we are only using the soluble portion for purification, this showed that we were losing a vast amount of our protein just because of the size of complexes they were forming. So to address this problem, we decided to try to modify or limit the ability of the complex formation to create complexes that were smaller, with the hopes that if the complexes are smaller, they would become more soluble and we'd be able to actually purify more protein. So that led to our next project, where we created a RIC called HLZE that has the same model antigen that I talked about previously, a small portion of the Zika virus. The first construct had the full-length epitope tag marked E, and the second one had a mutated epitope tag marked D. The epitope tag was mutated by shortening it so that it weakened the binding between the antibody and the epitope tag. This created, or we assumed that this would create smaller complexes. When we tested the amount of yield we were able to get with both these constructs, we found that the small change of limiting the size of the complexes greatly increased the expression levels. We went from 0.22 milligrams per gram of leaf fresh weight to 1.5 milligrams per gram of leaf fresh weight. However, another question that arose is that now that we are probably creating smaller complexes, we're able to increase expression levels. Do these two constructs still have the same immunogenicity? Because after all, our goal is to form these complexes so that we can trigger all the immune pathways and start generating antibodies against the antigen. So we did a mouse immunization study and directly compared these two constructs. On the graph over here, endpoint titers, you can see that we compared the antigen alone, marked ZE3, to the ZE, which is the full-length epitope RIC, to ZD, which is the short epitope. What we found is that the results for both of the RICs were highly similar with no statistical difference, and both of them were higher than ZE3 alone. This showed two things. First, it showed that the complexes, although small enough to become more soluble, were still enough to trigger the immune pathways that we were hoping to stimulate. And in addition, presentation of the antigen on the RIC platform increased its immunogenicity. So the outcome of this project was increased solubility without compromising the immunogenicity. Now that we have talked about how we can modify the RIC platform in order to enhance its versatility, both by increasing its solubility, keeping its immunogenicity the same, and also presenting two different fusion strategies for increased presentation of different antigens, we next moved on to studying the co-delivery of recombinant immune complexes with virus-like particles. The main goal for this project was to replicate some studies that we had done with the previous student. And we had found that if you were to display a certain antigen on both a RIC and a virus-like particle and co-deliver it, there was an increase, a synergistic immunogenicity that we saw. And our question was that, is this something that we can repeat with a different antigen, or was it something specific to that project? And so my job was to try to repeat that experiment, but use a different antigen to see if you were still able to get this synergistic response. So we created some different constructs. So the first construct was our standard recombinant complex. You have an antibody fused on the C-terminus to an antigen, the same Zika domain tree antigen, fused to an epitope tag. Then we co-deliver this with a virus-like particle. So virus-like particles are another type of vaccine platform that are non-infectious particles that have spikes on the surface. And each spike is a place where you can fuse an antigen. So when you are able to create these VLPs, you get a repeated pattern array of antigens, and this triggers different immune pathways. And so our thought process was that if you were to co-deliver a RIC with a VLP, you might be able to trigger different parts of the immune system and get a better response. We also tested whether or not we could see the same response if we were to create the N-terminal fused RIC with Z3 amount on the N-terminus of the antibody. So we created these two constructs and tested both groups. We did the same mouse immunization study, and what we found in the box marked red is that the co-delivery of VLPs with RICs, either C-terminal RIC or N-terminal RIC, resulted in increased antigenicity when compared to the VLP alone group and the RIC group. So that showed us a couple things. First, it showed us that the fusion site did not matter in this enhancement of immunogenicity. It showed that we were able to replicate the immunogenicity because both the co-delivery groups had higher responses to the VLP and the RIC. And all of the groups were much higher than the negative control of just PBS. So now that we've talked a little bit about my work on the recombinant immune complex platform, how we can use it to create vaccine candidates, how we can obtain a synergistic immune enhancement by co-delivery of RICs and VLPs, both delivering the same antigen, I'd like to change focus now to talk about how we can use immunoinformatics to design a vaccine. So most of my work has focused on trying to develop an ideal vaccine platform itself. But I'm also interested in how we can develop better antigens in order to display them on the platform. And so the latter part of my presentation will focus on some of the work that I've been doing to use computational tools in order to predict good epitopes that we can use on the RIC and VLP platforms. So I'm going to first start by going over the difference between a subunit versus an epitope-focused vaccine. What we have been working on so far have used subunit vaccines, where you deliver a subunit or a portion of the virus, such as the Zika virus domain tree antigen, and we present that on either the RIC or the VLP. This image over here is a space-filling model of the Zika virus domain tree antigen. However, you could create another type of vaccine where you focus on the epitopes. Now, once again, epitopes are the targets that are specifically recognized by B cells. So if you were to create an epitope-focused vaccine, instead of presenting the entire Zika virus antigen, you can focus only on these epitopes. And I've marked these two epitopes on the subunit vaccine as an example. So it's the exact same protein, either a space-filling model or in a ribbon diagram, and these are the two epitopes that I marked. So in an epitope-focused vaccine, you would only present the epitopes in the vaccine. And the goal of that would be to make sure that the immune response is producing antibodies that recognize those specific epitopes. But how would you come up with these epitopes? Well, before we talk about discovering epitopes, we first need to talk a little bit about T-cell activation. Till this point, I've been talking about B-cell activation, the generation of antibodies. However, there's a whole other arm of the adaptive immune system called T-cells. T-cells are a very vast and complex topic, but the only part that I'm going to focus on are CD4 T-cells, which are up here in the corner. And the reason why I care about CD4 T-cells is because they're responsible for helping their CD4 helper T-cells that help to activate B cells. And this activation helps B cells start generating large amounts of antibodies. So it's crucial that our B cells get proper T cell help. And in order to do that, we need appropriate stimulation of these CD4 T cells. In brief, we have antigens that come from the outside of the cell. They get taken up into the cell. And when they're inside the cell, they get loaded on these things called MHC class 2 receptors. MHC class 2 receptors take in these little peptides, which are short pieces that come from the pathogen, and they first get assembled, peptides to receptor, and then the receptors then get sent up to the cell surface. If the T cell recognizes the peptide, it binds the peptide and gets activated, and this activation then allows the T cell to go off and start helping B cells produce antibodies to the antigen. So my work has focused on trying to identify these T cell epitopes that would be useful for inclusion in vaccine candidates. The problem that I'm tackling is norovirus. Norovirus is the leading cause of acute gastroenteritis, and there are over 680 million cases every year. A vast number of these cases occur in children below the age of 5, and there's both significant mortality, which is death caused by the virus, and morbidity, which is sickness caused by the virus. There's also a huge economic impact because there are 680, the cost of norovirus annually is $60 billion, which is a large sum of money, all caused by this virus. Unfortunately, there is no licensed vaccine for norovirus, and current vaccine candidates have limited cross-strain protection. This is due for two main reasons. Number one, norovirus is a very diverse group of viruses, includes over 200 strains, and the problem is that infection with one strain does not provide enough protection to protect the host against infection with another strain. So there's been a lot of challenges in trying to create a vaccine that is able to broadly protect against multiple strains with good efficiency. The second problem is that there has been a historic inability to study the way the norovirus replicates. It has been difficult to culture norovirus in the lab, and this inability to understand how it functions has hampered vaccine development for a long time. So in order to create my vaccine, first we need to do a little bit of structural biology of norovirus. Norovirus has 3 open reading frames. The first one codes for non-structural proteins. Till date, non-structural proteins have not been analyzed at any great depth for inclusion in vaccine candidate. And so while my personal project primarily focused on VP1, which is generated by the open reading frame 2, my future plans for this project include studying some of these non-structural proteins to see whether or not they can be included in my vaccine candidate. VP1 is the major structural protein of the norovirus capsid, the shell that actually forms the virus particle. And VP2, which is generated by open reading frame 3, is the minor capsid protein. Both of these, VP1 and VP2, come together, and then they form the overall virus particle for norovirus. For this project, I primarily focus on VP1 as a starting point. And VP1 is the dimer. There's the shell domain, which is marked in orange, and there is a protruding domain. The protruding domain is then subdivided into VP1 and VP2. VP2 is the most variable of all the domains, and it's usually used as a way to determine which strain norovirus belongs to. VP1, however, is a very interesting domain because it contains neutralizing epitopes, and there have been some preliminary work that has shown there are conserved epitopes on VP2 that can provide cross-strain protection. So my goals for this project are to use immunoinformatics to identify epitopes that are broadly conserved across the different genogroups and strains of norovirus, and then create and test an epitope-focused vaccine that takes these epitopes that I have found through computational methods and then display them on the RIC and VLP platforms. So here is my overall blueprint of how I started my project. First, at the very top, I obtained VP1 sequences from the 3 genogroups that infect humans. There are 10 norovirus genogroups in total, but only G1, G2, and G4 infect humans. So I isolated all the VP1 sequences that I was able to obtain for these 3 genogroups, and then I cleaned them. So when I cleaned them, I made sure to remove any sequences that are only partial sequences, that were incomplete in any way, or had ambiguity, in which the researchers had uploaded the sequences weren't sure about the specific amino acid sequence for the protein, and so they used non-canonical amino acids to show their ambiguity. So I removed all of those, so at the end of the day, I only had full-length, clean VP1 sequences. Next, I used a tool from the immune epitope database in order to predict high-affinity T-cell epitopes. So the program just identifies the potential T-cell epitopes from the protein sequence that you give it. My interest, though, is only to pick the ones that are high affinity, which means that they are expected to have a high affinity between the peptide and the MHC2 receptor, because strong binding between the peptide and MHC2 receptor will help to better activate the T cells. And once again, T cell activation is important because the T cells, activated T cells, can go and stimulate the B cells to start generating antibodies. Once I had obtained a list of potential T cell, high affinity T cell epitopes, I then conducted consecutive conservancy analysis against G4 and G2 sequences. So I did this in order to test whether any of the epitopes that I had analyzed from the G1 were able to be conserved across the other T gene groups. And what I found is that there were 47 high affinity epitopes that were obtained with a maximum conservancy ID of 80%. So while we weren't able to obtain epitopes that were 100% conserved, we're at least able to obtain those that were 80% conserved. The next thing that I did is I did a similar analysis only for B cell epitopes. Remember, B cell epitopes are the epitopes that are recognized high affinity by antibodies, because my end goal is to create a vaccine that has both T cell epitopes and B cell epitopes. Okay. Once I had obtained all these epitopes, my next goal was to put them on a protein model so we can actually see where the epitopes were located. So for this protein model, I used the VP1 sequence, used a protein modeling software in order to create the model. And then on the ribbon diagram, which you can see, at the bottom are marked all the T cell epitopes that I obtained. So the T cell epitopes are marked on the ribbon diagram, and interestingly, all of them were in the shell domain, which is the bottom part of the VP1 protein. The B cell epitopes are marked on the space filling model in red, and as you can see that there are potential B cell epitopes all around VP1. I'm going to focus particularly on the B cell epitopes that are found in P1, because as I mentioned previously, experimental studies have shown that P1 contains neutralizing epitopes. And so my goal is to further study these epitopes in order to identify the best match of conserved T cell epitopes and B cell epitopes, link them together, and then express or present them on both the RIC and VLP platforms in order to create a vaccine that is designed to provide broad protection across multiple norovirus strains. In addition, I plan on studying those non-structural proteins so we can create the best vaccine and create a more broadly protected norovirus vaccine. So in conclusion, my PhD work has focused on three main aspects. First, enhancements to the RIC platform to increase its versatility. Next, the synergistic response that is seen upon the co-delivery of RICs and VLPs. And then finally, immunoinformatics in order to better design vaccine epitopes that can then be displayed on our RICs and VLPs. I would like to thank my committee for all their help in my PhD project, Dr. Hugh Mason, Dr. Qiang Chen, Dr. Tsafrir Mor, and Dr. Melissa Wilson. All the members of the Mason and Mor labs, both current and former members, our collaborators in the Chen lab, everyone from the animal care immunization team, because they handle all the mice immunization, the serum collection, spleen collection, everything else, and of course, my funding sources. And with that, I'd be happy to take any questions. Okay, so I can, if you'd like to put your questions in the chat box for everyone who is online, I can answer them as in one. And are there any questions from the in-person audience? Yes.
Great presentation, Mary. I had a question regarding the RICs. So we know that like some of them we can control like the sizes of the complexes. So what are the downsides of that when you inject it to the organism?
A large complex?
Possible, yeah. If they form like larger inside the organism, what are the possible, do you know?
Yeah, so the question was, what are the possible downsides of forming two large RIC complexes? There are a couple of downsides. First of all, the ideal complex should be between 20 to 200 nanometers, because that is the size that it can be efficiently drained into the lymphatic system and end up in the lymph nodes. So complexes that are larger than that can have some trouble being broken down into smaller components that can actually be taken to the lymph node for proper T cell and B cell activation. So that is one concern. The second concern is that if you form two large complexes that aren't broken up, they can start collecting in different parts, especially joints. That is called type 3 hypersensitivity. However, that is probably not a concern for our RIC platform, because we anticipate our RICs being broken down and presented in the immune system. But ideally, we want to create smaller complexes because smaller complexes have been shown to more efficiently activate immune pathways such as the Stephen-Key pathway and Fc gamma receptor pathway, and they would be easily taken into the lymphatic system and end up in the lymph nodes. Okay, from Caitlin. Well, when you're working with validating the RIC VLP platform, why did you stick to using only viruses? That's a great question. I initially started working with Zika virus as part of my undergraduate work on creating a chimeric monoclonal antibody that recognizes the conserved fusion loop. And we found that the Zika virus in particular was a perfect model for antigen for our work. Since first, the Z3 protein is able to be, is small enough to be easily expressed on our RIC platforms, and it also is weakly immunogenic. So we were able to show that you can take a weakly immunogenic antigen, mount it on our platforms, and greatly enhance its immunogenicity. And most importantly, we had collaborators in Dr. Chen's lab that had live Zika virus. So whenever we did a mouse immunization study, we were able to follow up our study of the mouse titers and also see whether or not they had neutralizing potential, whether they were able to neutralize live virus. And so we just had a good system, a good model of system in Zika virus. And then we just happened to work with other viruses that were herpes simplex virus, dengue virus, and other viruses from the flavivirus family mostly. However, you could probably mount various bacterial pathogens on our RIC platform. We just haven't tested them yet. Any other questions? Okay, yes, Caitlin, go ahead.
If I talk, can you hear me?
Yes, I can hear you.
Okay, so if you fuse an antigen before or after expression, like after with a cross-linker, would that affect the immune response?
That's a good question. If you were to cross-link an antigen after expression, so if you were just to produce the antibody and then cross-link an antigen, you have to make sure that the antigen also had the epitope tag that was recognized by the antibody, and that would enable it to form the immune complexes. But as long as you were able to keep the overall structure of a RIC the same, where you have an antibody with an antigen followed by an epitope tag that is recognized by the antibody, I think you should be able to create the same type of immune response that we see. Well, before everyone has to leave, I just want to thank everyone for attending virtually, for coming to my presentation, both online and in person. I'll still be around in case anyone else has more questions. If not, then I think I can move to my private defense if the rest of my committee is okay with that.